Hello, good evening, and welcome to this week's webinar by Technion UK. I'm Paul Charney, Chairman of the Technion UK, and I'm honoured to be able to host Professor Ester Segal. She is leading a multidisciplinary research group at the Faculty of Biotechnology and Food Engineering at the Technion. Professor Segal also serves as the Technion's Deputy Executive Vice President for Research. You know, women scientists are leading groundbreaking research across the world, but despite their remarkable discoveries, women still only represent about 33% of researchers globally, and their work rarely gains the recognition it deserves. But tonight, we are hugely proud to host one of those famous researchers. The Technion have always been trailblazers for generations of women. In 1924, at a time when academia was closed off for women in many countries, Technion's first class of 17 students included one female. Today, the percentage of women undergraduate students at Technion is 37%, graduate students 32%, and doctoral students is at 44%. This number continues to rise. The Technion's commitment to equal ratios and empowering women in the fields of STEM is a huge honor and pride for Technion in Israel. Also highly noted is Professor Segal, who was named among the top 50 most influential women in Israel last year. Tonight, Professor Segal will be talking about detecting disease in a fast manner and outside of the lab. So, without further ado, thank you so much, Professor Segal, for joining us tonight. Please feel free to ask questions in the chat box and enjoy yourselves. See you soon. Thanks.
Good evening. Happy Purim, Sophie.
Happy Purim.
My name is Ester Segal. I'm a professor here at the Technion at the Department of Biotechnology and Food Engineering. And together with me is Sophie Graham. Sophie is a PhD student. Sophie, introduce yourself.
Hello, everyone. My name is Sophie. I'm a PhD student on a dual doctoral program between the Technion under the supervision of Esti and the Leibniz University in Hanover, Germany, under the supervision of Professor Thomas Schipper. And I've conducted all of my degrees at the Technion at the Faculty of Biotechnology and Food Engineering. And that's it. Here I am.
So, Sophie, let's start talking about moving diagnostics from the lab to home. I got my second COVID-19 dose of the vaccine, of course. Did you get it?
Well, actually, no, because I was already sick about 4 months ago, which was not a very pleasant experience, not from the symptom side and not from the diagnosis. So this is very relevant to our talk today. So just I'll tell you my own story. I started to have my symptoms and until I got diagnosed, it was about 5 days where I was already with my family. So how does it work usually today? We have to go to, usually this diagnosis is carried out by taking a specimen from the patient. We go to the clinic, then we have to wait in the line. This is the corona line. And then our sample is taken. This sample is sent to a laboratory where it is studied by different analytical methods. And the main limitation is that those methods are lengthy, and this procedure is lengthy, and it usually takes about several days or weeks until we get those results. From my own experience, I had to wait about 5 days until I got my approval to go for the test, and then do the test and then wait for the results. So this was a very stressful experience.
I can only imagine.
Yes. So what we want to do, we want to develop a point-of-care diagnostics. And what does it mean? So point-of-care diagnostics are a test that could be performed near or by the patient himself without sending the sample to a centralized laboratory. So it can be just like a pregnancy test. This is the best example, or a blood glucose monitor, which are used today routinely. So this is just making a test outside of the lab. And just imagine that one day we could go to the farm store and find on the shelf near the pregnancy tests various other tests that could diagnose other diseases. And those are being constantly developed. For example, this mini-care assay for diagnosis of a heart attack. And if we talk about coronavirus, the best example would be, and this is the most relevant example today, we have this portable and personal PCR kit, or this paper-based immunoassay, which is actually very similar to a pregnancy test. So now maybe Esti would like to describe the technology that we do.
So I will give just a general background of what we do in the lab, and Sophie will then give an example of what she was specifically working on during her PhD. So we in the lab develop sensors, and sensors are basically very simple devices that one can take a urine test, a urine sample, or a blood sample, and just put them on the device, as you can see here in this schematic. And the device itself, using the technology that is integrated in the device, can diagnose and detect if we have a certain disease. And it broadcasts the signal to let's say a cellular phone, and then to a cloud, and then us, the patients at home, can know after very few minutes if we are sick or healthy. For example, in the case of COVID-19, if we should stay at home in lockdown, or whether we could go outside and meet our family. So Sophie, maybe you can tell more details about your PhD work. What type of sensors were you working on?
Yes. Okay, so I will elaborate about it more. So first of all, what are biosensors? These are small analytical devices which aim to detect specific molecules utilizing a bioreceptor, which is responsible for recognizing this target molecule, and it integrates this with a transduction system, which converts this binding of the target molecule into a measurable signal. And if we speak about medical diagnostics, so the target molecules will be biomarkers, molecule biomarkers for diseases. This could be a protein or an antibody or an RNA molecule, as in the case of COVID-19. So what is our technology? We use porous silicon as for the biosensing systems. And this porous silicon is a small chip, which its structure is kind of like a sponge. It has a very small holes inside, nanometric holes, and you can see how it looks like under a very high magnification of an electron microscope. And the porous silicon, we synthesize this kind of chips in our laboratory, and the fabrication is very simple. It also has very unique optical properties that we can use to fabricate optical biosensors. So how does that work? We illuminate the porous silicon with white light, and we measure the light that is reflected from the porous silicon. And if we have molecules that go inside into those nanometric holes, they will cause a change in this light that is reflected, and we will see this change in our with a very simple experiment, with very simple instrumentation. So my PhD work started with the aim of developing a biosensor for early diagnosis of pancreatic cancer. Pancreatic cancer is one of the most deadliest cancer types. It has the lowest survival rate. This is mainly because it has asymptomatic. It is asymptomatic at its early stages, or it has very general symptoms, which can be attributed to many other conditions. And this leads that most of the patients are diagnosed at a very late stage, when the tumor is already unresectable. So this leads to the poor prognosis. So we wanted to develop a biosensor that will allow its early diagnosis of this type of cancer. And we focused on a protein cancer biomarker, which is secreted at a very early stage of this disease, actually at the premalignant lesions, which is the stage just before the cancer appears. So you can imagine that this would be an ideal stage for early diagnosis. So we wanted to, I forgot to say that this protein is secreted to the pancreatic juice, and we wanted, this is the fluid that is secreted by the pancreas, and we wanted to detect this cancer biomarker, protein cancer biomarker in this type of fluid. But it's not simple because this protein is present at very low concentrations in these fluids, which contains millions of other molecules, and all those molecules will go inside our porous silicon, our silicon sponge, and they will cause a change in the optical signal that we measure. So this is not good, and you can think about it as finding a needle in a haystack, but not as nice as you can see here. It looks more like this. This is not very. This is a major challenge. So what do we do about it? We bind to the porous to our silicon sponge, recognizing molecules, capture probes, which are responsible for recognizing the specific molecules that we are interested in. In this case, this protein biomarker. And these molecules will specifically bind our cancer biomarker, and all of the other molecules will just flow around and won't bind to our silicon sponge and won't affect the signal that we get. And this indeed allowed us to detect this protein biomarker in a pancreatic juice. So just a few words about pancreatic juice. This is the fluid that is secreted by the pancreas, and it can be collected by endoscopy. Although it is relatively invasive, this fluid, because it is in close proximity to the pancreas, it has higher concentrations of all of the molecules that are shed by the tumor, and higher concentrations compared to those molecules in the blood. So while those molecules can be missed in the blood, we have more chances to detect them in the pancreatic juice. And this kind of biosensor could be well integrated in a standard endoscopy studies or in smart pills, and this could have a very good high impact. And this fluid is very complex. It's basically just a soup of proteins. It contains more than 500 different proteins, which makes it hard to detect this specific biomarker that we are looking for. But still, our biosensor could successfully detect our target protein biomarker. So you can see, this is demonstrated in this graph. It shows you the light that is reflected by our silicon sponge, the optical signal, for this pancreatic juice, positive for the protein biomarker, compared to the same fluid that is negative for the biomarker. And you can see that the positive fluid gave a significantly higher signal, while the protein biomarker is outnumbered by more than 100-fold of other proteins in these fluids. So this, in terms of the specificity, the ability to detect the specific protein in these complex fluids, we could do it successfully. So what was the challenge? This was in terms of the sensitivity. Usually, protein biomarkers are present in very low concentrations in those kind of fluids. However, our biosensors are able to detect those proteins at a 1,000-fold higher concentration. So we cannot really reach the specific concentration that we need to go to the clinic. And this took my whole PhD to understand what limits our biosensors and what can we do about it to improve their performance. So I first started from understanding what limits those biosensors. And for that, we derived a mathematical model that describes all the fundamental phenomena that lead to the capture of the target molecule in our silicon sponges. And this is how it looks like. So it is as scary as it looks, but it's good that I'm an engineer, and I had to use my engineering skills to do it. So I won't scare you with all of these equations. I will go to the nice images. So this mathematical model that we developed allowed us to understand what limits our biosensors and what do we need to do to improve their capabilities, both from the side of the biosensor design and also applying strategies to improve sophisticated strategies to improve their performance. And what strategies did we apply? For example, integration in microfluidic platforms. So what is microfluidics? Microfluidics is the technology that process small amounts of fluids in small channels with dimensions of tens to hundreds of micrometers. And the miniaturization, those systems miniaturize the device, and this miniaturization leads to significant advantages. For example, the consumption of the reagents is reduced. The time for the assay is also this is faster assay. Reduces the costs, and most importantly, we can integrate in our device several different functionalities and create kind of a lab on a chip. And to make it even better, we also worked on the integration of our biosensors with 3D printed microfluidics. So 3D printed technology is a rapidly advancing technology in recent years, and it makes this whole process of the microfluidic fabrication digital. So you design your device, your dream device in a computer, you then press a button, and about 2 hours later, or even less, you have your solid design in your hand. So we worked very hard on the integration of our silicon sponge in those microfluidic platforms, of those 3D printed microfluidic platforms. And this is how the final devices looks like. So they are those devices. This is just an example for the design that we use, but now we can exploit the 3D printing and design what we want and print it. And what do we do next? So, for example, now we work on developing a microfluidic biosensor, microfluidic platform that will allow us to simultaneously detect several different disease biomarkers at the same time. So this is very important for medical diagnostic applications, because usually one target molecule is not sufficient to have a reliable diagnosis. So in what else do? We also we just started a new collaboration with the gastrointestinal malignancy unit in the Rambam Hospital in Haifa, to develop a platform for detection of pancreatic cancer cells. So not proteins, in this case it's cells from blood, in order to allow its early diagnosis of this cancer.
Because actually we realized that we were a bit naive in the beginning of your PhD work, because we thought that detecting only one biomarker will allow us to detect pancreatic cancer with high accuracy. So what we are working now, advancing Sophie's technologies, is to detect other cancer biomarkers more accurate, and bring our technology to another level. And actually, I would like to describe another project in our lab, the project that Talia and Sin are working on in recent years. So I'm switching here to a completely different topic. We are not talking about cancer anymore. We are talking about antibiotics and about bacterial infections. I'm sure that most of you heard or know that we are, in fact, fighting antimicrobial resistance. We are fighting bacteria that are basically called now superbugs, and no antibiotics can actually tackle this bacteria. And also in the case of COVID-19, if we want to bring the relevancy of this bacteria to the era of COVID-19, it was actually found recently, Sophie, that 1 out of 7 patients that are hospitalized due to COVID-19 actually acquired a very dangerous secondary bacterial infection, and some of them actually die as a result of a bacterial infection and not as a result of the initial COVID-19 virus. So next slide, Sophie. What we did in recent years is developed a very small device, basically based on the same silicon technology that Sophie described to you. And here you can see a cassette which integrates the individual silicon chips that Sophie just described. And if we try to dive into what happens inside such a device, is that we have bacteria that are, the source of these bacteria is from, let's say, our urine or infected blood. The device can attract and attach these bacteria, target bacteria cells into the structure. And as a result, as Sophie described, we see a change in the way the light is reflected back from the device. So you can imagine that you have this device. In case of urine, you just, sorry to say, you just pee on it, Sophie. And you see a change in color when bacteria is attached to the silicon chip, and we can learn if we have a bacterial infection or not. And this is not the most important part. The most important part is that, Sophie, if you turn to the next slide, is that not only we can say if you have a urine infection or not, we can also direct your doctor or physician into what is the best antibiotics that can treat your infection. Because as I mentioned earlier, many of these bacteria are already resistant to many antibiotics that exist. So the doctors needs to know what antibiotics he should prescribe you in order, first of all, not to lose time and get you the best clinical result, right? And the second important thing is the fact that because there is such a huge overuse and misuse of antibiotics, many of the bacteria are getting more and more resistant. So in a global perspective, if a doctor will know in advance what antibiotics he should prescribe to you, it will lower the rate and emergence of new resistant bacteria. So currently, if you have a urine infection, for example, or even a sore throat, and you want to know if you are infected and what antibiotics the doctor should prescribe to you, one just take a sample, and the clinical sample is sent to a centralized lab. In the lab, they use sophisticated instruments. There are many lab technicians that process your sample. And in the case of bacteria, it takes at least 2 days until you know if you are positive for the bacteria, and it will take possibly another day if the doctor would like to know which antibiotics he should give you. So 2 to 3 days, it's not realistic if you want to know and to treat your patient in a rapid manner. So what we've developed is this device, as I showed you earlier, and basically in less than 90 minutes, the device can tell you if you are infected or not infected, and more importantly, which antibiotics and at what dosage the doctor should prescribe you. So this is really a game-changing technology. And here I show some of our collaborators and students who actually do the work. So first of all, I would like to mention Dr. Sagi Spritz. Dr. Spritz is a neurologist from the Bnei Zion Medical Hospital, and it's remarkable that during the COVID-19 fight, he used to come after shifts into our lab and take off his coat and wear another one and work on these devices. And this is Sin and this is Talia. And you can see that Talia is standing next to a prototype device that we built and can be already deployed in the hospital. So clearly this is a very important technology. And so what's next after diagnosing rapidly urinary tract infections? We are now starting another project where we want to advance this technology even further. And this goes to treating sepsis. So I don't know if everyone knows what is sepsis. Sepsis is a case where you have a bacterial infection in the blood, and this is a very dangerous and deadly situation. If you look on this graph, you can see on the y-axis the survival rate, and on the x-axis you can see time. So if more than 6 hours pass, the patient, the survival rate of patients suffering from sepsis is decreasing to only 30%. So time is very crucial to treat this patient. So if the doctor makes a mistake and give a sepsis patient the wrong antibiotics, in many cases it would mean that the patient will die as a result of sepsis. So what we are doing is taking the urinary tract infection technology that we just developed into fighting sepsis. And I hope that next year, maybe next Purim, we will be able to show that in less than 60 minutes, less than 1 hour, we can detect sepsis and direct the physician how to treat this sepsis next to the patient bed already in the ER without even sending the sample to the lab. So this brings me almost to the end of our presentation and discussion today. I would like to thank all my group members, especially our collaborators, Dr. Raul Kolodner and Dr. Sarel Alachmi from the medical centers around Haifa, my group members, some of those who are missing in the picture, and of course, many thanks to the funding agencies that allows us to do these very important studies and researchers. I suggest, Sophie, that we will move to the question session. Let's see if there are questions from the audience. Okay, we are getting a lot of questions, Sophie. And apparently the first one is to you, Sophie. So let me read the question to you and let's see if you can answer it. So, Sophie, what is next in your career? So I guess our audience is very interested to learn what are you going to do after your PhD at the Technion.
Yes. So I'm looking now for a postdoctoral position. I would very much like to proceed my career in the academy and to open my own lab one of the days. So I'm now looking for a postdoc, in fact, in the UK. I would very much like to do my postdoc in Cambridge. So hopefully next year I will be joining United Kingdom.
Maybe you can eat Ozen Aman. I don't know how you say it in English. Next time with the audience in the UK.
Yes, hopefully.
So, and the second question is also to you, Sophie. So, how does the Technion encourage female students to engage in leading roles in the academy and industry? I think that I can also answer it, but the floor is yours.
Well, in fact, I didn't really feel any difference between a female and male. In the Technion, in our faculty, we have almost the same ratio of female scientists and male scientists, so this was not really an issue. But generally, there are a lot of scholarships that encourage women, specifically for postdoctoral studies. Yes, so this is, there are a lot of support.
So I have to say personally, Sophie, that I would like to see you joining the Technion after your postdoc position in the UK and opening a new lab. And there is another question. Let me see. So the next question is, can you describe other projects in your lab? So I think that we are about to run out of time. So I will just say that we are working not just on medical diagnostics, we are also working on therapy. So one recent project that we just published was a project where we designed a silicon chip, basically very similar to the chip that you worked on, Sophie, for diagnostics. We designed a chip that can be injected and implanted in the brain, and it can release very important drugs and therapeutics. Specifically, the work that we just published deals with the Alzheimer's disease and the fact that many patients that are suffering from Alzheimer's disease are lacking very important proteins in the brain that are responsible for the brain function. So what we demonstrated for the first time is that you can take this silicon chip, you can implant it in a sick mice, in this case, and the protein degrades the protein is released from the chip while the chip itself degrades in the brain. It doesn't stay in the brain of the patient, and it releases this very important protein in a timely manner. So this is one, I think, very important project that goes on in our lab. Another question that we just got is, how did the situation caused by the COVID-19 affect your research? I think clearly everyone already understood that Sophie was sick. We were in a race to get the vaccines. The lab was closed. So during the 3 lockdowns that we had in Israel, the lab was opened and closed for different times. Some of the students were sick. Some of the students had to stay at home with their young children. And it was, it actually still is. I can't say that we are
not over yet.
It's not over yet. And for sure, research has slowed down significantly. And I really hope that with the help of science, we can move forward and beyond the COVID-19 pandemic and promote our research even further. So, Sophie, I would like to thank you for joining me today.
Thank you for inviting me.
And I would like to thank the audience for joining us. I send my regards to all of you from Israel, from the Technion. And I hope that next year, in next Purim, we will be able to join you live.
Yes.
So thank you very much, everyone.
Thank you.